Hi, my name is Daniel Hornburg. I'm a principal scientist with Seer, and today I will present to you our Proteograph, a multi-nanoparticle platform that enables plasma proteomics at scale and speed, significantly improving performance versus traditional deep proteomics methods. But why do we need new ways to interrogate the proteome? Well, take for example blood plasma, which comprises signals from most tissues and is a rich source for protein biomarkers for disease detection. However, its large dynamic range, spanning more than 10 orders of magnitude, is a huge challenge for any downstream detector. That means the depth of coverage achieved with measuring neat plasma is limited and does not provide the whole picture. More complex workflows like depletion and peptide fractionation get deeper but trade off throughput, scalability, coverage, and precision. At Seer, we use specifically engineered nanoparticles to address that challenge. When nanoparticles are introduced into a physiological environment such as plasma, proteins and other molecules form a so-called protein corona. The quantitative composition of the corona depends on the physicochemical properties of the nanoparticles, such as their size, shape, charge, hydrophobicity, as well as on the properties of the proteins. This provides a unique avenue to deal with the dynamic range challenge. Nanoparticles bind proteins based on affinity, and a single nanoparticle binds a broad range of proteins across the entire dynamic range. By using a panel of diverse nanoparticles, we can interrogate the entire complexity of a proteomic sample. To maximize the effectiveness of nanoparticles, we have developed a solution called the Proteograph that simplifies and automates this process and integrates it into an existing mass spec workflow. To assess the performance of the Proteograph pipeline, we used a single plasma pool and compared the 5 nanoparticle panel, neat and depleted plasma, as well as a traditional deep plasma proteomics workflow that includes depletion and peptide fractionation. All samples were run with the same short 30-minute gradient. 5 nanoparticles yielded significantly more protein groups, in particular covering the low abundant proteins at higher precision than a deep fractionation workflow. In our recent Nature Communications publication, we employed 5 nanoparticles to interrogate a clinical cohort. We took 141 subjects to investigate their plasma in less than 2 weeks to a depth of 2,500 proteins. We then used that rich source of information to build a classifier to distinguish between cancer and controls. As you can see here, the classification performs pretty well, and we were able to identify novel and previously described potential protein biomarkers. If you want to learn more, join our Hupo Connect webinar, have a look at our Nature Communication publication, follow us, or join our growing cross-disciplinary team. Thank you and stay curious.